Hi everyone, my name is Stephen Buchert at Cal State East Bay, and today I'm going to be talking about the effects of sleep on behavioral decision making and reproductive fitness in the fruit fly, also known as Drosophila melanogaster. Who feels like they got enough sleep last night? I know I typically don't. But this is really important because sleep affects our memory, our cognition, decision making, hormones, even our immune system. But how much sleep do we actually need? Well, sleep is variable between species. Different animals all have different requirements of the amount of sleep they need. And sleep is also variable within species. Even in humans, the amount of sleep we need is different depending on our age. So the question becomes, is there an advantage to sleeping more or sleeping less? To answer this question, I'm going to be using the fruit fly, also known as Drosophila melanogaster. This is an excellent model organism for this question because it has a sequenced genome. It has pretty simple genetics with many human homologs. It also has a very quick and simple life cycle, which makes experiments very easy. And it has sleep very similar to humans. It has circadian rhythms and a diurnal cycle. I'll be looking at this question using a line that I call the long and short sleepers, also known as the DGRP line. This line has been inbred over many generations, with the longest sleepers being bred with the longest sleepers and the shortest sleepers being bred with the shortest sleepers. This resulted in very distinct sleep phenotypes, with long sleepers sleeping much more than normal and short sleepers sleeping much less. Now, before we could begin with the actual experiments, we first had to make sure that the flies that we got matched the flies in existing data. We did this in two ways. We used a video sleep rig and also a Drosophila activity monitor. The video sleep rig uses background subtraction in order to track the flies visually, and the Drosophila activity monitor uses an infrared beam to track when they cross. Now we measure sleep as 5 or more minutes of inactivity, and we recorded the flies for 3 days. We also used 2 different temperatures because their behavior could change depending on the temperature, and our incubator was set to a 12-hour day, 12-hour dark light cycle. The results of our sleep experiment were pretty consistent with the existing data, and we found 3 lines of significantly different short sleepers and 3 lines of significantly different long sleepers compared to the normal groups. We had to find a way to determine reproductive fitness in Drosophila, and we found that luckily enough they have a very visual reproductive dance, which involves a singing aspect marked by a unilateral wing extension to a 90 or 45 degree angle. This was very beneficial to us because in our experiments we were able to record them and then mark this visually. In our experiments we also used virgin flies, and we had to sex them male and female. Sexing the flies can be done visually with a microscope by looking at their size and shape, as well as their coloration. Virgin flies can also be found by a dark spot on their abdomen called a meconium. This is the first fecal matter from when they are a larva going into a pupa and emerging. It won't be passed for a few hours, which is also the amount of time that it takes the fly to mature before courtship or reproduction. We chose these virgin flies because they'll be more vigorous in courting, which gives us more data to count. We put the flies in what we call courtship wheels, which are 8-chamber wheels with separate chambers for the males and females that are combined at time of recording. We took 20-minute videos and looked for unilateral wing extension, which is a choice behavior of the Drosophila, marking whether it wants to eat, sleep, or court. This is what it looks like in the courtship wheel, and here's a short video showing what it would look like. This is sped up, but here's an example. We can see G here is very vigorous in his courting, and he's really not giving up. The results of our courtship experiment found one line that courted significantly more than all of the other lines, the DGRP38 line. Now we wanted to see how courtship choice behavior affects reproduction. To do this, we used 24 well plates, and we mated 48 mature female flies with 20 males for 3 days. We then separated them and placed 2 female flies in each well for another 3 days to lay their eggs, at which point we counted them. which is very tough because fly eggs are pretty small. Here's an example of the fly eggs. You can see they're very small. We definitely had to use microscopes for this, and it was pretty tedious. But we did it, and here are our results. We found that again the short sleeping high courting line 38 laid a significantly high number of eggs, but also that the long sleeping line 335, which is a normal courting line, also laid a significant number of eggs. So in this research, we found that sleep amount affects courtship choice behavior in males. We found this with our DGRP38 results. We also found that courtship deficits in long sleepers may be compensated for by female flies. We found this with our 3 through 5 results. The long sleepers still laid significantly more eggs, which must be compensated for by female flies. We also see that sleep amount in courtship does not determine reproductive fitness. We know this because the short sleeping line and the long sleeping line had significantly more eggs than normal. In the future, we want to explore phenotypic plasticity between sleep and egg laying. This just means how the environment is affecting these behaviors within these genotypes. We would do this by looking at mate competition, which I like to call a love triangle. We would take a short and long sleeping male and put them in with a normal female. And we would see how this situation would affect their choice between eating, sleeping, and courting. We also want to look at courtship compensation to see which sex is responsible for the compensation that we saw where the long sleepers still laid significant a number of eggs. I want to thank Dr. Divya Sitaraman for allowing me to take over this project and teaching me so much about flies and research. I also want to thank Pamai Murakami and all of the others at the Fly Lab who helped me with this project. Does anyone have any questions?